Now that we have our primers attached to our sequence, what we're going to do is use benchling to tell us the size of our PCR products. So remember that the first thing we really needed to remember about PCR was that it's going to copy the DNA between the two primers, and that is it. And so to predict the product of a PCR, what you're going to do is simply measure the distance between the two primers. So this is primer 23. This is primer 24, right? And so the DNA that will be copied will be added onto these primers. So we're going to include the amount of sequence in the primer itself and measure in between. The easiest way to do this is to click one primer, hold shift, and click the other primer, and it will highlight the DNA sequence in between. So when we actually do this PCR using primer 23 and 24 in the presence of E. coli 16S rRNA sequence in our genomic DNA, that is the PCR that should happen, right? Because we're going to amplify in between the two primers. And the other most important thing about PCR was that it's only going to work if the primers actually match, right? This primer matches 23, this primer matches 24, this reaction should work. So while you have the sequence highlighted, down here at the very bottom, this gray bar along the bottom is going to say bases, which is the length of the entire sequence that you're looking at. So 1541 is the whole thing. What we have highlighted is here from start at 5 to end at 191 for a length of 187 bases. So we know that the PCR product from E. coli with primers 23 and 24 should be 187 bases. And so you would plot that right here in your table, right? Primer pair number 1, which is primers 23 and 24, in the E. coli gDNA should be 187 base pairs. And you'll just do that for all your combinations, right? So for E. coli, we can now try the next one, right? So here's 23 and 25. So if we highlight there, this is 529. So you would put that here for 23 and 25 as a pair, 529. What you can't do is 25 and 24, right? Why? Both of these primers are pointing in the same direction, right? They're both reverse primers. So we're going to add DNA right to left off of this primer, add DNA right to left off of that primer. We end up with two identical strands that kind of overlap instead of two complementary strands, right? We need to copy the top strand and the bottom strand to be able to make double-stranded DNA as a PCR product. So that's why you always have to use 23 as a forward and pair it up with one of the other ones. That's a reverse primer. Looking around our E. coli sequence, those are the only primers that match, right? The algorithm didn't find any more primers. So that means primer 26 and primer 27 don't match the E. coli sequence. And so you would not get a product. You would just say none if you ran that PCR, or you shouldn't, right? Because that primer doesn't appear to stick anywhere in this sequence. Yes, we did use rather strict parameters when we looked for primers, so that's what we think should happen, but we'll see what happens in real life. On the topic of strictness of parameters, let's look at that one weird one. So if you, on your staff epi sequence, you'll see that primer number 23 is highlighted in red. It's just telling you like, hey, this isn't a perfectly matching primer. And you can see there are two incorrect bases in here. Because they are not right next to each other and they're not directly on the 3' end of the primer, this primer should still work. We might have to lower our annealing temperature when we do the PCR to make sure that it works. But it should still be able to base pair well because every single other base is going to base pair. And we've got, you know, a G and C and more G's and C's over here on this end where we're going to add bases. So it's most important this end is attached. So it should work just fine. All right, so measure all your distances for all of your primer pairs and see what you get. Again, if that primer is not there, then you can expect it to not bind, and therefore your PCR will produce nothing. So you can indicate that.